I want you to try something for me. Right now. Just stop.
Stop what?
Breathing.
Oh, we're starting with the high stakes immediately.
Yeah, just hold it. Don't take that next breath. I was actually doing this earlier today while I was, reading through the research for this deep dive. I got to about maybe 45 seconds before my brain just started absolutely screaming at me. It is a very primal panic.
It really is. It's the most immediate, visceral connection we have to staying alive. I mean, you can go days without water, you can go weeks without food, but you cut off this one invisible gas for 3 or 4 minutes, that is game over.
And yet, we do it unconsciously. You're doing it right now while listening to us. 12 to 16 times a minute, awake or asleep. And because it is so automatic, I think we tend to trivialize it. We think, oh, breathing, air goes in or air goes out. Simple.
Yeah, and calling human respiration simple is, well, it's perhaps the greatest insult you could pay to evolutionary biology. What you felt, that rush of air hunger when you held your breath, that's just the alarm system of an incredibly complex machine. It's a machine that involves fluid dynamics, heavy-duty chemistry, structural engineering, and really precise neural regulation.
It's a machine that somehow manages to extract a specific gas from the atmosphere, transport it to billions of individual cells, and simultaneously remove a toxic waste product, all without you having to lift a finger. So today we're going on a mission. We are taking a deep dive into the unit on breathing and exchange of gases.
And looking at our stack of notes here, this is heavy on the source material from the NCERT biology text, specifically Chapter 17. So if you are listening and prepping for Neet or really any medical entrance exam, you might want to grab a pen.
Okay.
We are going to hit all the high-yield stuff.
All right, but our goal isn't just to, read you a list of definitions. We want to unpack the actual logic of this system. We want to move from just rote memorization of volumes and pressures to understanding why the body's built this way.
We will cover the whole journey today. We'll start with the evolutionary perspective, looking at how different animals solve the breathing problem. Then we will look at the hardware, which is the human anatomy. We will break down the software, basically the physics of how we pull air in.
Yeah, we'll crunch the numbers on lung volumes, which I know is a huge favorite topic for examiners. We'll get into the nitty-gritty chemistry of gas exchange and transport, the blood bus, as I like to call it. And finally, we'll look at the control center and what happens when the whole system breaks down.
It's a very comprehensive look. So let's just start at the very beginning. The biological mandate. Why do we breathe? I mean, really, why?
Well, the simple answer is to get oxygen. But why do we need oxygen?
It's all about energy, right? Precisely. As we have seen in previous deep dives into cell biology, every organism, from a simple sponge to a complex human, needs energy to perform activities. To get that energy, they have to break down nutrient molecules, things like glucose, amino acids, fatty acids.
Catabolic reactions. You break the bonds, you release the energy.
And the most efficient way to break those bonds in those carbon-based fuels is by using oxygen. It is oxidation, but there is a catch here. When you burn that fuel using oxygen, you produce a byproduct. Carbon dioxide.
And carbon dioxide isn't just some useless filler. It is actually harmful if it builds up.
Yes. It is toxic in high concentrations. It physically changes the pH of your body fluids, turning them acidic. So the problem every single animal faces is twofold. You need a continuous supply of oxygen coming in to burn the fuel, and you need a continuous removal system for the carbon dioxide waste.
Input and output. Oxygen in, carbon dioxide out. And this process of exchange, taking oxygen from the atmosphere and swapping it for the carbon dioxide produced by the cells, is what we formally call respiration.
Now, here is where it gets absolutely fascinating. The goal is always the same, right? Oxygen in, carbon dioxide out. But the method, the engineering solution, that changes dramatically depending on where the animal lives and how complex its organization is.
It's like evolution just keeps redesigning the ventilation system based on the size of the building.
Exactly. Let's look at the simple creatures first. Lower invertebrates like sponges, coelenterate, flatworms. Do they have lungs?
I would assume not. I mean, a sponge is basically just a porous blob sitting in the water.
Correct. They do not need lungs. For these animals, the entire body is in direct contact with the water. They exchange oxygen and carbon dioxide by simple diffusion over their entire body surface.
So they literally just breathe through their skin, well, their whole surface area.
Essentially, yes. The distance the gas has to travel is short enough that simple diffusion works fine, but there's a hard limit to that design. As animals get bigger or thicker, the cells in the middle of the body get too far away from the outside world.
Because diffusion is slow.
Oh.
You can't wait for oxygen to just passively drift 3 inches deep into your body. You would be dead before it got there.
Exactly. So once you get past a certain size, you need a transport system. Take the earthworm. I am sure you have seen them out on the sidewalk after it rains.
Oh yeah, they're always super slimy.
That slime is literal survival. Earthworms use their moist cuticle, their skin, for respiration. The moisture is crucial because gases must dissolve in liquid to diffuse across a membrane. If an earthworm dries out, it suffocates.
Okay, so moisture is the key there. But what about insects? Because they seem way more active than worms. They are flying, buzzing around, jumping. They must have a high metabolic rate, which means they need a ton of oxygen.
They definitely do. And their solution is one of the most unique in the entire animal kingdom. They do not use blood to carry oxygen like we do. If you squash a bug, the fluid isn't red, right? It does not have hemoglobin for gas transport.
So how on earth does the oxygen get to the leg muscles of a grasshopper?
They have a complex network of tubes called tracheal tubes. It is like a dedicated ventilation system embedded directly into their body. These tubes have openings on the surface of the insect, and they branch out to transport atmospheric air directly to the cells and tissues.
That is wild. It's like having a dedicated air conditioning duct going into every single room in the house instead of just cooling the main hallway and hoping the cold air spreads.
That's a very good analogy. It is highly efficient for small sizes. But when we move to vertebrates, the strategy splits based on the environment. It comes down to water versus land.
Right, because if you are in water, like an aquatic arthropod, a mollusk, or a fish, you are dealing with dissolved oxygen.
And for them, the evolutionary solution is gills. We call this branchial respiration. Gills are these specialized, feathery, highly vascularized structures that are designed to pull dissolved oxygen directly out of the water. They maximize surface area in a fluid medium.
But gills don't work on land. We all know this. A fish out of water dies.
They die because the gills stick together without the buoyancy of the water to support them. That drastically reduces their surface area and they just dry out. So for terrestrial forms, which includes amphibians, reptiles, birds, and mammals, evolution developed vascularized bags called lungs. This is known as pulmonary respiration.
Pulmonary meaning lungs. Now, I do have to ask about amphibians, like frogs, because they live in both worlds.
Frogs are the ultimate multitaskers of the respiratory world. They have lungs for breathing air, yes, but they can also respire through their moist skin, which is called cutaneous respiration.
So it's a built-in backup system.
A very vital one. It allows them to breathe while they are completely underwater or when they are hibernating deep in the mud. They essentially act as the bridge between the earthworm's skin strategy and the human's lung strategy.
Must be nice to have options. So we have gone from simple diffusion to moist skin to tracheal tubes to gills and finally to lungs. Which brings us to the main event today, the system designed for high-energy terrestrial living, the human respiratory system.
The human hardware.
Let's trace the path of a breath of air. I really want to visualize this. I am imagining I am an oxygen molecule. I'm just floating outside, and then suddenly, suction. I get pulled in.
You enter through the external nostrils. You travel through the nasal passage directly into the nasal chamber. And notice something immediately. The environment changes. It is not just an empty tube.
It's lined with hair and mucus, right?
Yes. This is the conductant part of the respiratory system, and it has a very specific set of jobs. It filters out foreign particles like dust and pollen. It humidifies the air, adding essential moisture, and it brings the incoming air to body temperature.
Which is really important. Nobody likes breathing freezing cold, dry air in the middle of winter. It physically hurts your lungs. So the nose is basically the lobby where you get warmed up and cleaned off before entering the main building.
Exactly right. From the nasal chamber, the air moves down into the pharynx. Now, the pharynx is a somewhat tricky intersection. It is the common passage for both food and air.
The danger zone. This is where choking happens.
It is. But we have a brilliant safety valve. The air moves from the pharynx into the larynx, which is the cartilaginous box which helps in sound production, hence the name, the sound box. But right at the opening of the larynx, there is a region called the glottis. And covering that glottis is a thin, elastic, cartilaginous flap called the epiglottis.
I just love that word, epiglottis. It sounds like a Greek villain, but it's actually the hero of the story.
It is the master traffic controller. During swallowing, the epiglottis covers the glottis. It folds down like a protective trapdoor. This prevents food from entering the larynx. It ensures your lunch goes down your esophagus to your stomach
Yeah.
and not into your fragile lungs.
And if that flap fails, even for a fraction of a split second, we cough violently. That is just the body saying, intruder alert.
Correct. Now, assuming our oxygen molecule has safely passed the epiglottis, it enters the trachea, the windpipe. This is a straight tube extending all the way down the neck and into the mid-thoracic cavity.
I want to pause on the trachea for just a second. If you touch the front of your throat, you can actually feel those hard ridges.
Those are cartilaginous rings. But here is a critical detail that shows up on exams and is functionally essential. They are incomplete rings. They are C-shaped.
Incomplete? You mean they don't form the full circle around the pipe?
No, they are open on the dorsal side, the back side.
Why is that? Why not just have a solid, complete pipe? Wouldn't that be a lot stronger structurally?
Think about what sits directly behind the trachea, the esophagus, the food tube. When you swallow a large, poorly chewed bite of food, the esophagus needs room to expand. If the trachea were a rigid, complete circle of cartilage, the esophagus would have nowhere to go. You would have serious trouble swallowing.
Oh, wow. So the C shape keeps the airway open so it doesn't collapse like a wet straw when you inhale, but the open back allows the esophagus to bulge slightly into the tracheal space when you swallow food. That is incredibly smart design.
It really is. Now, let's follow the trachea further down. It does not go on forever. It reaches a very specific anatomical point in the chest where it splits.
The bifurcation.
Yes. The trachea divides into a right and left primary bronchi. And for the anatomy students listening, you need to know exactly where this anatomical split happens. It is at the level of the fifth thoracic vertebra.
T5. That is the landmark to remember.
Correct, T5. Above that vertebra, you have one single pipe. Below that, you have two.
Okay, so we have split into the right and left bronchi. We are formally entering the lungs now.
And this is where the tree analogy really comes into play. The trachea is the main trunk. The primary bronchi are the thick main branches. Then within the lungs, the branching goes almost fractal. Primary bronchi divide into secondary bronchi, then into tertiary bronchi, and then into thinner and thinner tubes called bronchioles.
And finally, we reach the very end of the line. The terminal bronchiole.
Right, and at the very end of those tiny twigs, the terminal bronchioles, we find the leaves of the tree, the alveoli.
The alveoli. These are the real stars of the show.
They are. They're very thin, irregular-walled, and highly vascularized bag-like structures. Vascularized means they are completely covered in a dense net of blood vessels. This is the actual site of gas diffusion. This is where the outside air finally meets the inside blood.
And there are millions of them, right?
Millions upon millions. If you were to somehow flatten out all the alveoli in your lungs, they would cover roughly the size of a tennis court. That massive surface area is precisely what allows us to absorb enough oxygen to power a large human body.
So the lungs themselves are basically just a massive spongy collection of these branching tubes and alveolar bags, all packed tightly into the chest.
Correct. And the lungs are covered by a double-layered protection system called the pleura, or pleural membrane.
2 layers. What is sitting in between them?
Pleural fluid. It acts as a vital lubricant. The outer pleural membrane is in close contact with the thoracic lining, which is your chest wall. The inner pleural membrane is in direct contact with the lung surface itself.
It's kind of like having 2 smooth sheets of glass with a thin layer of water in between them. They stick together tightly, but they slide past each other perfectly.
That sticking together is the key. It means that when the chest wall moves, the lungs absolutely must move with it. And that concept leads us perfectly into the third part of our dive, the physics of breathing, the actual mechanism.
This is where we need to talk about the sealed box concept.
The thoracic chamber. It is an airtight chamber. Dorsally at the back, you have the vertebral column. Ventrally at the front, the sternum. Laterally on the sides, the ribs. And on the lower side, you have the dome-shaped diaphragm.
It's completely sealed off, and that seal is everything. Because, and this honestly surprised me when I first learned it years ago, we cannot just muscle our lungs open.
No, we cannot. We have absolutely no muscles attached directly to the alveoli. You cannot just flex your lungs to draw air in.
So how do we actually get the air in?
We use the physics of pressure gradients. Air, like any fluid, naturally moves from an area of high pressure to an area of low pressure. To get atmospheric air to rush into the lungs, which is inspiration, the pressure inside the lungs, the intrapulmonary pressure, must be less than the atmospheric pressure outside.
We basically have to create a vacuum inside our chest.
Exactly. We need to generate negative pressure. And we do this by expanding the size of the box. Remember Boyle's law from chemistry.
Volume goes up, pressure goes down.
Precisely. So how do we increase the physical volume of the thoracic chamber? Two main muscle groups get to work. First, the diaphragm.
That large dome-shaped muscle right at the bottom.
When the diaphragm contracts, it flattens out. It actively moves down. This movement increases the volume of the thoracic chamber in the anteroposterior axis, that is top to bottom.
So the floor of the box basically drops down.
Second, the external intercostal muscles. These are the specialized muscles located between your ribs. When they contract, they physically lift the ribs and the sternum up and slightly outward.
Kind of like lifting the handle of a bucket.
Yes, the bucket handle motion. This increases the chamber's volume in the dorsoventral axis, front to back.
So the floor drops, the walls expand outward, the sealed box gets significantly bigger.
The overall thoracic volume increases, and because of that strong pleural seal we just talked about, the lungs are physically pulled open. The pulmonary volume increases.
And per Boyle's law, since the volume inside the lungs just went up,
the pressure inside drops. It falls below the standard atmospheric pressure, and the air outside, which is currently at a relatively higher pressure, just rushes in naturally to fill the newly created space.
we don't really suck the air in at all. The universe just pushes it in.
That is a somewhat poetic way to put it, but physically it is accurate. That is inspiration. It is an active process requiring actual muscle contraction and energy.
Now, what about letting the air back out? Expiration.
Normal expiration is largely a passive process. You basically just stop doing the work. The diaphragm relaxes and passively arches back up into its normal dome shape. The intercostal muscles relax, and the heavy ribs and sternum drop back down to their resting positions.
The box naturally shrinks back to size.
The thoracic volume decreases. That means the pulmonary volume decreases. The pressure inside the lungs rises slightly above the atmospheric pressure, and the air is pushed back out.
It's just the natural elastic recoil of the whole system, like letting go of a stretched rubber band.
Exactly right. Now, we can force it if we want to. If you are blowing up a balloon, you use your abdominal muscles to actively push the diaphragm up much harder. That is active or forced expiration. But normal, quiet breathing, just relaxation.
And we repeat this cycle 12 to 16 times a minute. And doctors can actually measure this moving air using an instrument called a spirometer.
Which brings us to the numbers. Part 4 of our dive, the respiratory volumes and capacities.
I always feel like this is the part where biology students tend to zone out, but we need to make this stick. Because these numbers tell a massive story about how our lungs functionally work.
And for medical exams, this is absolute high-yield territory. You must know these definitions and normal values.
Let's play a bit of a guessing game with the listener. First one, tidal volume. TV. This is your completely normal, chilled-out breath. How much air are we talking about here?
It is approximately 500 milliliters.
So roughly the size of a standard small water bottle. Or a normal can of soda plus a little sip.
Yes. That is the volume that quietly moves in and out with every single resting breath. Over the course of a minute, considering our breathing rate, that is roughly 6,000 to 8,000 milliliters of air.
Okay, now suppose I take that normal, quiet breath, but then I decide to just keep inhaling. I want to fill my lungs to the absolute maximum until I cannot fit another single molecule of air in.
That extra volume of air you can forcefully pull in after a normal inspiration is called the inspiratory reserve volume, or IRV.
How much is that reserve on average?
It is surprisingly large. It averages about 2,500 to 3,000 milliliters.
Oh, so my extra reserve is like 5 or 6 times the size of my normal breath.
Correct. It really shows you just how much capacity you are holding back for emergencies or for strenuous exercise when your body demands way more oxygen.
Okay, let's go the complete opposite way. I breathe out normally, then I push. I squeeze the abdominal muscles. I push all the air out that I possibly can.
That extra air you forcefully expire after a normal expiration is the expiratory reserve volume, ERV. That averages about 1,000 to 1,100 milliliters.
Okay, so I've pushed out all my ERV. My face is probably turning red at this point. My lungs are completely empty now, right?
No. And this is a vitally important concept. You can never completely empty your lungs of air. If you somehow did, the delicate alveoli would collapse and physically stick together, and the tissues would be severely damaged. There is always some air left behind.
The unexpellable air.
Exactly. We call it the residual volume, RV. It averages roughly 1,100 to 1,200 milliliters.
So even after I blow out my birthday candles, with every ounce of strength I have got, there is still over a liter of air just sitting in my chest.
Precisely. And that remaining air is absolutely crucial. It ensures that gas exchange can continuously occur even while you are in the middle of breathing out. Think about it. If your lungs went completely empty, the oxygen flow to your blood would just stop for a second during every single breath cycle. The residual volume keeps the bridge open constantly.
That's a really great point. It smooths out the entire supply chain.
Hmm.
Now, the textbook takes these individual volumes and combines them into what they call capacities.
Yes, pulmonary capacities are just specific sums of two or more volumes. They are highly useful for clinical diagnosis of lung issues.
Let's run through them quickly. Inspiratory capacity, IC, the total amount I can breathe in.
That is your normal breath, the tidal volume, plus your maximal reserve, the inspiratory reserve volume. So TV plus IRV.
Expiratory capacity, EC, the total amount I can forcefully breathe out after a normal breath in.
That's your normal breath out, tidal volume, plus the extra forced amount, the expiratory reserve volume. TV plus ERV.
Here is a slightly trickier one, functional residual capacity, FRC.
Think carefully about the name itself. Functional residual. It is the volume of air that normally remains in the lungs after a normal, unforced expiration. You are just sitting there, you breathe out normally. What is left inside the box?
Well, it would be the air I could have forced out if I tried, which is the ERV, plus the air I absolutely cannot force out, the RV.
Correct. FRC equals ERV plus RV. This is the volume of air sitting inside your lungs right now as you listen to us talk.
We have two big ones left. Vital capacity, VC.
This is defined as the maximum volume of air a person can physically breathe in after a forced expiration. Or, viewed the other way, the maximum volume you can forcefully breathe out after a forced inspiration. It represents the total usable range of your lung system.
So that is basically everything in the tank except for the untouchable residual volumes. It's ERV plus TV plus IRV.
Exactly right. And finally, the easiest one to remember, total lung capacity, TLC. This is the absolute total volume of air the lungs can hold when fully inflated.
The whole tank.
Yeet.
Vital capacity plus the residual volume. VC plus RV.
You got it. All the volumes summed up together.
Okay, so we've got the air into the system. We know exactly how much is sitting there. Now we need to do the actual deal. Part 5, the exchange.
The exchange of gases. This critical swap happens primarily at the alveoli, but we also have to remember it happens later between the blood and the body tissues.
And the main driver here is incredibly simple, right? There's no biological pump pushing the gas across. It is just basic diffusion.
Simple diffusion. And as we know, diffusion is driven purely by pressure or concentration gradients. A gas will naturally move from an area of high partial pressure to an area of low partial pressure.
We really need to look at the partial pressure table from the text. This is another vital set of numbers that tracks the gas as it moves through the system. I like to imagine a waterfall. Things just naturally flow downwards. Let's track the oxygen molecule first.
Okay. Out in the atmospheric air, the partial pressure of oxygen, which we write as PO2, is 159 millimeters of mercury. That is the very top of our waterfall.
We breathe that air deep into the alveoli.
In the alveoli, the PO2 drops to 104 millimeters of mercury. It's slightly lower than the raw atmosphere because the fresh air is mixing with that residual volume we just talked about, the stale air sitting in the lungs. But 104 is still a very high pressure.
Now, let's look at the blood arriving at the lungs. The deoxygenated blood returning from its trip around the body. What is the PO2 in that returning blood?
It is a low 40 millimeters of mercury.
Whoa, 104 sitting in the alveoli, just 40 in the arriving blood. That is a very steep hill to roll down.
It is a massive gradient. That huge difference in pressure drives oxygen to rapidly rush from the alveoli across the thin membrane and directly into the blood. It happens almost instantaneously.
So the blood loads up on oxygen. It becomes fully oxygenated blood. What does its PO2 rise to now?
It rises up to 95 millimeters of mercury. This rich oxygenated blood then travels through the arteries to all the body tissues.
And what is the partial pressure of oxygen inside those active body tissues?
It is down to 40 millimeters of mercury again, because the cells in the tissues are constantly consuming oxygen to burn fuel and make energy.
So now we have another gradient. 95 in the arriving arterial blood, 40 in the needy tissues, another downhill slope. Oxygen jumps off the blood bus and flows into the cells.
Precisely. It is all beautifully driven by physics. Now let's look at the return trip. carbon dioxide.
Okay. The tissues are constantly making CO2 as a waste product, so the PCO2 in the tissues must be high, right?
Yes, it sits at 45 millimeters of mercury.
And in the fresh oxygenated blood arriving at those
The
tissues.
PCO2 there is only 40.
So carbon dioxide moves from the tissues at 45 into the blood at 40. The blood now becomes what we call deoxygenated blood. It travels all the way back to the lungs. The PCO2 in that returning deoxygenated blood is 45.
And waiting in the alveoli, the PCO2 is 40 millimeters of mercury.
Wait, hang on a second. 45 in the blood to 40 in the alveoli. That is only a pressure difference of 5. The oxygen gradient we just talked about was huge. 104 down to 40, that's a difference of 64. The carbon dioxide gradient is tiny, just 5. How does the body get the CO2 out fast enough with such a weak push?
That is a highly perceptive question, and the answer lies entirely in the specific chemistry of the gas itself. It's about solubility.
Solubility, like how well it dissolves.
Exactly. The solubility of carbon dioxide is 20 to 25 times higher than the solubility of oxygen.
Wow, 25 times. So it dissolves much, much easier.
Exactly. Because of that chemical property, it simply does not need a huge physical push to cross a liquid barrier. It slips through the membrane almost effortlessly. This means that even with a tiny pressure difference of just 5 millimeters of mercury, carbon dioxide can diffuse out of the blood and across the membrane incredibly fast.
That perfectly explains it. Speaking of the membrane itself, the barrier these gases actually have to cross, what is that structurally made of?
It is formally called the diffusion membrane. It consists of 3 distinct microscopic layers: the very thin squamous epithelium of the alveoli itself, the endothelium of the alveolar blood capillaries, and the basement substance situated right in between them.
3 layers. That actually sounds pretty thick for a gas to cross quickly.
It sounds thick when you list the components, but the total thickness of all 3 layers combined is much less than a single millimeter. It is an ultra-thin barrier specifically optimized to facilitate that rapid, continuous exchange.
So we have successfully exchanged the goods. Oxygen is in the blood, CO2 is out. Now we need to physically transport them around the body. Part 6 of our dive, transport of gases.
The blood bus. Let's look closely at how the passengers are actually seated for the journey.
Let's do oxygen first. How does it travel through the bloodstream?
The vast majority, about 97% of all oxygen, is transported inside the red blood cells, the RBCs. Specifically, it is chemically bound to hemoglobin. The remaining tiny 3% just travels in a dissolved state in the blood plasma.
So hemoglobin is the main carrier here, and that's an iron-containing pigment, which is why blood is red.
Yes. Oxygen binds to the iron in hemoglobin in a reversible manner to form a complex called oxyhemoglobin. Each individual hemoglobin molecule can carry a maximum of 4 molecules of oxygen.
Now, there is a graph in the textbook here that basically haunts every biology student, the oxygen dissociation curve. It plots the percentage saturation of hemoglobin against the partial pressure of oxygen, and it makes a very specific shape.
A sigmoid curve, an S shape.
Why is it S-shaped, though? Why wouldn't it just be a straight diagonal line? More pressure, more binding.
This is where the molecular magic of proteins happens. It visually represents a phenomenon called cooperative binding. Think of the hemoglobin molecule as a specialized taxicab with 4 locked doors.
Okay, a taxi with 4 seats.
When the taxi is completely empty, all the doors are tightly locked. It is chemically very difficult for that first oxygen passenger to pry a door open and get in. That struggle represents the flat, slow-rising bottom part of the S-curve.
It's hard to get the process started.
But once that very first oxygen molecule fights its way in and binds, it physically alters the 3D shape of the entire hemoglobin protein. It effectively unlocks the other 3 doors. Suddenly, it becomes incredibly easy for the second, third, and fourth oxygen molecules to just jump right in. That rapid loading represents the steep upward slope of the S-shape.
So the molecule is almost greedy. Once it gets a tiny taste of oxygen, it rapidly wants to fill the rest of the seats?
Yes, it chemically cooperates to load up. But the reverse process is also true, and this is vital for survival. The entire curve can physically shift left or right based on the local environment.
The shift. This is known as the Bohr effect, right?
Essentially, yes. Think logically about where you want hemoglobin to greedily grab oxygen and where you desperately want it to let go.
Well, I obviously wanted to grab oxygen tight when it is in the lungs.
Correct. In the alveoli, the local conditions are ideal for binding. You have high PO2, very low PCO2, a lesser hydrogen ion concentration, and a lower overall temperature. These specific factors all act together to shift the curve to favor the formation of oxyhemoglobin. It essentially locks the oxygen into the seats.
And I want it to let go when it reaches the deep tissues, especially if I'm using working muscles.
Exactly. In the active tissues, the local environment is totally different. You have low PO2, highly elevated PCO2 from waste, a high hydrogen ion concentration, meaning it is more acidic, and a higher physical temperature from metabolism. These conditions strongly favor dissociation. They literally force the hemoglobin protein to change shape again and dump its oxygen load.
It's honestly amazing. The physical waste products of a working tissue, the carbon dioxide and the heat, actually serve as chemical signals telling the blood, hey, we are working extremely hard over here. Drop the oxygen right now.
It is physiological efficiency at its absolute finest. And here's a concrete delivery stat you need to memorize for exams. Under normal physiological conditions, every 100 milliliters of oxygenated blood delivers around 5 milliliters of oxygen to the tissues.
Okay, 100 milliliters delivers 5 milliliters of oxygen. Got it. Now, what about the return passenger on the bus? Carbon dioxide.
Carbon dioxide is a bit trickier to manage. It actually utilizes 3 different modes of transport to get back to the lungs.
Let's hear mode 1.
Mode 1 is simple, just dissolved right in the blood plasma. About 7% of CO2 travels this way.
Fair enough. Mode 2.
Mode 2 is bound to hemoglobin. About 20 to 25% travels this way. It forms a compound called carbamino hemoglobin. Note that it binds to the amino group of the protein, not to the iron core like oxygen does. They don't directly fight for the exact same seat.
Okay, so that accounts for maybe 30% total. What about mode 3, the big one?
The vast majority of carbon dioxide, roughly 70%, is carried in the blood as bicarbonate ions, HCO3-.
Wait, bicarbonate? Like baking soda? How does a waste gas just turn into a salt ion?
It is an ingenious chemical disguise, and it heavily relies on a specific enzyme located inside the red blood cells called carbonic anhydrase. This enzyme is present in very high concentrations inside the RBCs and just in minute quantities in the plasma.
What exactly is the chemical reaction?
The enzyme takes carbon dioxide and water, CO2 plus H2O, and rapidly mashes them together to form carbonic acid, H2CO3. Then, almost instantly, that unstable acid dissociates into a bicarbonate ion, HCO3-, and a hydrogen ion, H+.
So at the tissue level, where CO2 waste is constantly piling up, the enzyme grabs the CO2 and water, converts them into bicarbonate, and basically hides it out in the plasma.
Yes. The CO2 is essentially trapped and disguised as a dissolved salt ion for the long journey back to the chest. This clever trick keeps the actual dissolved PCO2 gas levels in the blood relatively low, which ensures that even more CO2 can keep continuously diffusing out from the tissues down its gradient.
That is brilliant. And then what happens when this disguised bicarbonate finally gets back to the lungs?
The entire reaction just violently reverses itself. At the alveolar level, where the local PCO2 is very low, the reaction proceeds in the complete opposite direction. Bicarbonate enters the red blood cell, the enzyme turns it back into carbonic acid, and then splits it right back into CO2 gas and water. The disguised CO2 is suddenly released as a gas, diffuses across the membrane, and you just breathe it out into the room.
It's like the ultimate biological smuggling operation. Turn the gas into an invisible ion to transport it quietly, then turn it right back into a gas at the border to dump it.
It really is.
Right.
And the removal stat to pair with the oxygen delivery, every 100 milliliters of deoxygenated blood delivers approximately 4 milliliters of CO2 back to the alveoli.
Okay, so 5 milliliters of oxygen delivered, 4 milliliters of carbon dioxide removed per 100 ml of blood. Those are the golden numbers to remember.
Correct. Keep those written down.
Okay, so we are in the home stretch now. We've got the machinery completely understood, the plumbing, the physics, the chemistry. But who is actually driving the machine? Who's controlling it? Part 7 of our dive, regulation.
The biological control center. Unlike the human heart, which possesses its own internal pacemaker node to beat autonomously, breathing is heavily, almost entirely, controlled by the neural system. You absolutely need the brain to send a signal to tell the diaphragm muscle to contract.
Where exactly is the boss located in the brain?
Deep in the brainstem. Specifically, in a region called the medulla oblongata, there is a specialized, dedicated cluster of neurons there called the respiratory rhythm center. This is your primary regulator. It sets the basic, quiet pace of your life. In, out, in, out.
But obviously the pace changes. If I suddenly sprint up a flight of stairs, I breathe way faster. Who tells the main rhythm center to speed things up?
There is a sort of middle manager located slightly higher up in the pons region of the brain. It is called the pneumotaxic center.
Pneumotaxic. That almost sounds mechanical.
It effectively acts like an override switch or a brake pedal. A strong neural signal from the pneumataxic center can actively reduce the duration of inspiration.
And if you cut the inspiration phase short, you naturally have to switch to the expiration phase much sooner.
Exactly. Shorter, shallower breaths naturally mean faster breaths overall. So this center actively alters your overall respiratory rate based on the body's immediate needs.
Now here's the specific detail that completely blew my mind when I was reading through the source notes for this. What chemical information are these brain centers actually looking at to make these rapid pacing decisions? Because you would logically think they're constantly monitoring oxygen levels, right? Just making sure we have enough vital fuel.
You would naturally think so. It seems completely logical. Low oxygen, better breathe faster. But you would be entirely wrong.
Really? How so?
The actual role of oxygen in the regulation of a respiratory rhythm is physiologically quite insignificant.
Right. The body doesn't care about its own oxygen levels.
Not as the primary driver, no. It cares profoundly about internal waste. There is a highly specialized chemosensitive area situated directly adjacent to the main rhythm center. This area is exquisitely sensitive to carbon dioxide and hydrogen ions.
So we are basically running a pollution monitoring system, not a fuel gauge system.
Exactly. If carbon dioxide or hydrogen ion levels start to creep up in the blood, which naturally happens the moment you start exercising or burning more energy, this chemosensitive center activates instantly. It aggressively signals the rhythm center to alter the respiratory rate to quickly eliminate the toxic waste.
That is just wild to think about. When I am completely gasping for air at the end of a long sprint, feeling like I'm dying, it's not actually because my body is aggressively craving more oxygen.
Yeah.
It is because my brain is absolutely desperate to dump the accumulating carbon dioxide.
Precisely. The panic is a waste removal panic. There are also peripheral receptors located down in the aortic arch and the carotid artery that do the exact same job. They constantly monitor arterial blood for rising CO2 and hydrogen ions, and they send rapid warning signals up to the rhythm center. Oxygen dropping is only used as a very late, distant backup emergency signal.
All right, that is fascinating. Finally, let's wrap up with part 8, troubleshooting. Disorders of the respiratory system, because clearly with a machine this complex, things can break.
They certainly can. We will briefly cover the 3 main disorders specifically highlighted in the source text.
First up on the list is asthma. I think a lot of people are familiar with this one.
Yes. Asthma is characterized by a significant difficulty in breathing, which classically causes an audible wheezing sound. The underlying pathological cause is an acute inflammation of the bronchi and the smaller bronchioles.
So the actual tubes just get swollen and narrow. It is essentially a plumbing issue restricting airflow.
Correct. The pipes narrow, making the work of breathing much harder. Second on the list is emphysema. This one is mechanistically very different.
The text notes this is a chronic disorder.
Yes, it develops slowly over a long time. In emphysema, the delicate alveolar walls are physically damaged or destroyed. Remember our tennis court analogy for surface area?
We have millions of tiny bags providing massive area.
Well, emphysema basically destroys the thin walls separating those tiny bags. So instead of millions of tiny efficient bags, you end up with fewer, much larger floppy bags. This drastically decreases the total respiratory surface area available for gas exchange.
And less surface area immediately means less oxygen absorption into the blood, no matter how hard you breathe.
Correct. It is debilitating. And the major primary cause of this specific destruction is long-term cigarette smoking. It physically and chemically destroys the microscopic architecture of the lung tissue itself.
That's a grim reality.
Yeah.
And the third category I mentioned, occupational respiratory disorders.
This broad category heavily affects workers employed in specific industries like stone breaking, grinding, or mining. In these environments, so much fine particulate dust is produced that the body's natural upper defense mechanisms, all those hairs and the sticky mucus in the nasal chamber, they simply get completely overwhelmed.
So the dust makes it all the way down deep into the lungs. What happens then?
Long-term chronic exposure to these fine particles leads to severe deep tissue inflammation. And eventually this triggers fibrosis, which is the massive proliferation of rigid fibrous connective tissues. The lung essentially becomes stiff, scarred, and unable to expand properly, causing extremely serious and irreversible lung damage.
Just a strong reminder that workers in those conditions absolutely must wear proper, high-quality protective masks. It's not optional.
Absolutely essential for long-term survival.
So man, we have really unpacked it all today. From the simple, passive diffusion of a tiny sea sponge all the way to the incredibly complex chemical disguise of carbon dioxide hiding inside our red blood cells. It's so much more intricate than just air goes in, air goes out.
It is a truly remarkable biological system. The precise pressure gradients effortlessly created by the diaphragm muscle, the unimaginably massive surface area of the microscopic alveoli, the elegant chemical affinity and cooperative binding of the hemoglobin protein, it all works in this perfect, continuous concert just to keep us alive.
And just coming back to that very first thought we had about holding your breath, think deeply about the residual volume concept again, that liter or so of air that absolutely never leaves your chest.
Yes, the continuous mix.
It really means that every single breath you ever take is just a blend. You deeply inhale fresh atmospheric air, it rushes in, swirls around, and immediately mixes with the stale, used air that was already sitting down there in the dark. We are genuinely never truly completely empty, and we are never truly completely full of 100% fresh air. We are always just living our lives on a continuous biological blend.
It is a perfectly balanced dynamic equilibrium. It quietly ensures that the internal fire of life never flickers out, even for the brief split second occurring directly between your breaths.
I honestly think I'll never think about taking a simple deep breath the exact same way ever again.
And for all the students listening out there, specifically the ones grinding for Neet, my final advice is to go back to the book and really study the diagrams. Look closely at the cooperative binding on the sigmoid curve. Memorize the waterfall drops on the partial pressure table, because those visuals are the absolute keys to locking this complex information into your long-term memory.
Excellent advice. Thanks for taking this deep dive with us today. Keep breathing, keep studying, keep learning.
We will see you all on the next dive.
Catch you later.